Alright guys, so I just wanted to go over the Azocasein assay one more time. So here we go, Azocasein assay. Now, so we are doing this assay to evaluate the optimal trypsin cleavage efficiency, so optimal pH and temperature at which trypsin can function most effectively to cleave azocasein. So looking at trypsin, we know that trypsin is an enzyme, right? And it will cleave azocasein. So specifically, trypsin is a protease. So whenever you see ACE at the bottom, you can count on, first of all, that being an enzyme, but also it's commonly means that it cuts whatever is preceding it in that word. So in this case, protease, it's going to cut proteins. So commonly cuts, you could say breaks, digests, whatever it comes before that. So there could be lipases, could be proteases, etc. Now, trypsin specifically cleaves after certain amino acids. So we remember from the primary structure of proteins. Proteins are made of amino acids. So there are certain amino acids that trypsin will specifically cleave after, those being lysine. So that's denoted by the capital letter K, but also arginine. So trypsin will cleave after lysine and arginine. Now, how does this azocasein assay work? So azocasein is a modified milk protein. Keep in mind, modified milk protein. And how it's modified is it has these, what are called sulfanilamide groups attached to it. So let's just say I'm looking at a, I guess, primary structure of azocasein. It could just be any amino acids. Maybe there's some K's in here, an R. And azocasein will have all these sulfanilamide groups attached to its structure, basically. And that's important because sulfanilamide groups absorb light at 440 nanometers. So given all this information, we can create an assay out of this to determine the optimal temperatures and pH that trypsin works at, or trypsin functions best at, or can cleave azocasein most efficiently at. So in order to understand how this assay works, let's imagine a situation where trypsin can work as best as it can. So it can work under its optimal conditions. So this first will be the optimal conditions. Let's just say that we're just going to draw out what happens at trypsin's most optimal temperature and pH. So we would first have our microcentrifuge tube with whatever reaction buffer in there. We would have the azocasein in there, and then we would add our trypsin, right? So let's just say there's a certain part of let's just say there's a certain part of azocasein that has the amino acid sequence of something like this. So that's a certain part of azocasein. Remember, it has all these sulfonilamide groups, right? All over it. That are like an orange dye color. So they'll basically turn the supernatant orange. But more importantly, we said that trypsin cleaves after lysines and arginines. So it'll cleave 1, 2, 3, 4, 5. Now, remember, we also are going to add the TCA in there at one point. So the TCA, if you remember, TCA precipitates larger fragments, specifically larger fragments of protein. So it'll precipitate larger fragments of our azocasein. In other words, precipitate undigested azocasein. Or uncleaved, or parts of larger fragments that trypsin was not able to act on. So, for example, there's this larger fragment right here, right? So let's just say that TCA is able to precipitate fragments of 4 or more amino acids in length. It would precipitate this fragment. So after we add our trypsin to the azocasein and reaction buffer, trypsin would do its job. It would cleave at these right after these amino acid residues, these lysines and arginines, and we would get the resulting fragments. XXK, XR, XK, XR, that large fragment. Let's just add another, I don't know, K right there. And then there would be an XK, right? Now, what happens after this? So we would heat it or incubate it at whatever temperature. Then we add our TCA, right? So we add our TCA, which precipitates the larger fragments. It's going to precipitate this fragment, right? So then, and then we centrifuge. So TCA and centrifuge. And this is what we'll get. Remember, this larger fragment is going to be solid now, which is going to let us be able to centrifuge it out into the pellet, right? So there's going to be a pellet. TCA is going to make this solid or precipitate it into solid form. We'll centrifuge it. That will end up in the pellet. Whereas all these guys, all the other fragments, and then that XK at the bottom, they'll remain in the supernatant. Now remember, regardless, all of these still have sulfonilamide groups, right? They all still have their sulfonilamide groups over here. Even this guy still has the sulfonilamide groups, but that guy's now in the pellet. But when we're looking at just the supernatant up here, we can see that there are some sulfonilamide groups floating around attached to these free smaller fragments of amino acids. And that'll result in the supernatant being an orangish color, right? And that's what we take with the pipette, run on the spectrophotometer, grab the absorbance reading. Remember, this is optimal conditions, right? Trypsin can do its job most efficiently. It can cleave every single, or it can cleave after every single lysine and arginine. Then we'll take that supernatant, run it on the spectrophotometer, and get that absorbance reading. Now, let's imagine another situation. Maybe we are choosing an extreme temperature or an extreme pH where it'll actually affect negatively trypsin's ability to cleave azocasein. So let's just say we choose an extreme pH. Maybe, like, we know that trypsin functions, human trypsin at least, functions great at around pH of 8 in the small intestines. Let's say we choose an extreme pH of 1, okay? It's completely different than where we know it functions at normally. Not that 8 is the most optimal, it could be, but that's just where it's found. That's just where we find it. Let's say we choose an extreme pH of 1. We do the same thing, so we have the same azocasein, XXK, XR, XK, XR, 1, 2, 3, 4, KXK. Remember, it's got those sulfonylamide groups. Now, but because of this extreme pH, negatively affect, I think it's effect, yep, trypsin's ability to do its job. So what's going to happen if trypsin can't do its job? It's not going to be able to cleave as efficiently. It's going to miss some of these fragments. So let's say it misses that first one, but it can cleave here. Misses that one, can cleave here. Maybe it can cleave here and here. Now, maybe it can cleave here too, right? So up here we had a total of 6 cleaves. We had 4 cleaves down here because of the extreme pH. What's going to happen here? In our reaction tube, we have our reaction buffer at a pH of 1 now, right? These are going to be the resulting fragments. Remember, all these still have their sulfonylamide groups. We had our TCA and centrifuge it. So TCA will precipitate all the large fragments. In this example, I'm just assuming that it can precipitate fragments, again, of 4 or more amino acids. And we'll get what this time? We'll get a larger pellet because there's larger fragments here, right? So we'll get that top one and the bottom fragment in the pellet this time. Then up in here, we'll get the 2 small fragments. All still with their sulfonylamide groups. But now these larger fragments, more of the azocasein in general now is in the pellet, right? Now, comparing these two guys, which one's going to have the higher absorbance? This one. Because there's a lot more sulfonylamide groups in the supernatant than compared to this one. If we take this supernatant and run it on the spectrophotometer against this one, this has obviously a lot more sulfonylamide groups in the supernatant than compared with this one. This one will have the higher absorbance, which indicates higher trypsin efficiency, right? So that's how we're going to tell that's how we're going to tell which conditions are more optimal for trypsin to cleave azocasein at. I just realized there should be a K up here. So K for lysine. So what can we conclude from this? We can conclude that the higher the absorbance, the higher trypsin efficiency, and the more azocasein breakdown, right? And vice versa. The lower the absorbance, the lower trypsin efficiency, and less azocasein digested. Now, just a couple last minute things to think about here, guys. While looking at this, this is a very simple layout of what's really going on here. In reality, there would be thousands of azocasein molecules and thousands of trypsin molecules. All the azocaseins being broken apart, so it'd be a lot more convoluted here than what we're actually seeing. This is just a very simple example of what's going on with just one azocasein protein and one trypsin enzyme. Second thing to think about is that assuming that the reaction conditions such as temperature and pH are adequate for azocasein to stay in, azocasein would not be in a linear or primary structure form like this. It would in fact be in its 3D conformation or its tertiary structure. But it would still be fragmented by trypsin. Trypsin would still be able to cleave these lysine and arginine residues after those residues to fragment that protein. It would just depend on the reaction conditions, whether or not azocasein would be in a 3D conformation, or if, let's say, it was at a temperature and or pH that is well outside of azocasein's optimal conditions, then yeah, it would be possible that azocasein could denature into more of a linear form like this, or unfolded form, away from its 3D conformation. And the last thing I want to mention is that in this example, I am assuming, again, that TCA is capable of precipitating fragments of 4 amino acids in length or higher, whereas that very well might not be the case. I don't actually know what fragment lengths TCA is capable of precipitating, actually, but the concept, regardless, remains the same. So being that if trypsin is working more efficiently, it'll result in smaller fragments being produced, which will be then be found in the supernatant here, with more sulfonilamide groups in the supernatant. That'll then lead to a higher absorbance, indicating higher trypsin efficiency. Same thing, if trypsin is not functioning correctly, then it'll miss out on some of the cleavage sites that it's supposed to cleave at, for example, here and here, and which will then result in larger fragments, more quantities of larger fragments that TCA can then precipitate, and a lower overall absorbance because there will be less sulfonilamide groups in the supernatants and more of those larger fragments in the pellet there. You guys, if this video helped you out at all, please be sure to like and subscribe. It really helps out. Remember, subscribing does not cost anything. All it means is that On your YouTube homepage, it might just tell you if I put out another video. I hope this helps you guys out before you submit your presentations. Please reach out with any questions. Thanks.